So Chapter 11 discusses animal diversification, and we'll start, like we've been doing with most chapters, looking at a basic definition of what you need to have as far as characteristics go to be grouped in an animal category. So first of all animals are heterotrophic, which means they eat other organisms. So remember, troph is to eat something, so being heterotrophic means you literally eat others. So hetero is others, trophic is to eat, so they eat others. Also, all animals move at some point during their life cycle. Some animals might only move as larval stages, and then they settle in one location and grow into the adult stage, but at some point in their life cycle they move. And all animals are also multicellular. When animals are grouped into different categories, it's done on a variety of things, and if you're just looking at the development of the animal, the physical structures, kind of the phenotype of the animal, then you would use things starting with defined tissues, if they have actual layers of tissue or not, and we'll see things like sponges don't have defined tissues, but we move into jellyfish and they do. We also look at symmetry, and symmetry basically means how many ways could that organism be cut in half and both sides approximately match. So this jellyfish has radial symmetry. We could cut it in half in nearly any direction up here and both sides would be the same. With bilateral symmetry, by meaning two, bilateral symmetry, we can only cut it in half in one direction. So if we cut this poor little frog in half in this direction, the right and left sides would approximately match. But if we cut it in any other direction, those two halves would not match. So it has just bilateral symmetry. Another characteristic used to classify organisms occurs during embryological development, and that's very important when you're classifying things, to look at the embryo and how it develops. And this is looking at the digestive system and how that forms the hollow tube through the body for organisms. There are two types of digestive system formations in animals. The first is the protostome. Now, stome means mouth, and proto is first. So these are literally first mouths. So for protostomes, what happens, you have this ball of cells forming the body, and part of that starts caving in and pushing through the center, and for protostomes, this first opening becomes the mouth. And then as it breaks through on the other side, then that becomes the anus. So the head of the animal and the back of the animal. Deuterostomes, again, talking about the mouth formation, except in deuterostomes, the mouth forms second. So this first opening that forms as this tissue layer starts to cave in towards the middle, that first opening becomes the anus, and the second opening where it breaks through on the other side becomes the mouth. And most species that we'll talk about today in this chapter are protostomes. We'll only talk about two different phyla that are deuterostomes, including humans. So humans are actually deuterostomes. So your anus, when you were developing, formed before the mouth did. We also use growth patterns to classify organisms, looking at whether they molt or if they have an external skeleton that they have to shed, like insects, or if they have internal support to hold their body upright so they don't need to shed anything from the outside. We will go through and look at animals in several phyla, and we're going to go from fairly simple structures to more complex structures. So we'll first start, and this chapter has really nice figures that ask questions as you go along. So do they have specialized cells that form tissues? Well, sponges don't have any specialized tissues. So we'll look at sponges first. Cnidarians do have specialized tissues, and so do all the others. Then we'll look at the type of symmetry. We'll look at protostomes first, and we'll look at how they grow in the protostome group, and then we'll look at some deuterostomes as well. One thing I want to make sure to emphasize in this chapter, even though we're going from simple body plans to more complex, that complexity doesn't necessarily mean that it's better or more successful. Simply being alive means everything currently on the planet is successful, because the definition of not being successful on an evolutionary timescale is that you're extinct. So if you're not extinct, then you're able to find food, escape predators, and reproduce. So it's not necessarily a higher or lower organisms, which is kind of an old-fashioned terminology for referring to things. Everything is about equal on an evolutionary timescale because we're not extinct yet. However, if you compare certain species, we have this earthworm and the tiger. If I asked you which one was more complex, you would say the tiger obviously is much more complex than the earthworm. Which one is more successful? Well, they're both currently successful because they're both alive, but on the long-term time scale, the earthworm was around before the tiger in the fossil record, and chances are, based on current things occurring with tiger populations, the earthworm's going to outlast the tiger by quite a bit. So the most successful, looking at a long-term time scale, would probably be the earthworm, even though you'd be tempted to classify that as a lower organism because of its simple body plan. It actually is potentially more successful than the tiger. Now, if environmental conditions change dramatically, who knows, maybe the tiger would survive longer than the earthworm, but it's highly unlikely. In this chapter, we're going to look at only 9 of the 36 phyla. So we're not going to cover all types of animals. We're just going to look at 9, the 9 that you'd be most familiar with and that you'd encounter on a regular basis. Again, we're going to go through sponges, cnidarians, flatworms, annelids, mollusks, and then we'll go through roundworms, arthropods, echinoderms, and chordates. And they all branch from the same ancestral protists. We can see sponges were the first to branch off, so we'll look at those first. Also, this is broken down into invertebrates, so most of these are invertebrates. So everything from sponges to the echinoderms don't have an internal skeletal structure the way we do, and so they're invertebrates. It's not until we get to the chordates that we start to see the same structures that we have in our body, so the nerve cord and the backbone, things like that. So we'll cover the invertebrates first, and they make up the vast majority, and then we'll look at the chordates. First of all, sponges, they don't have any specialized tissues or organs. Basically, they are a set of cooperative cells that form these kind of open tower structures, and what they do is they filter feed. So they will, using cells that line up on the inside with flagella, these flagella will move and generate a current. So it pulls water from the outside of the body structure to the inside, and then it can move out the top. As that water passes through, there are specialized cells that catch any food particles. They filter their food right out of the water. And then to get that food to the rest of the body of a sponge, those catching cells pass on the food particles to kind of amoeba-like cells that crawl around the rest of the sponge body and share the food with all of the cells that aren't directly catching it. So that's how sponges are able to get their food and get it throughout their body. They just rely on cells that can crawl around and share the food. They also have free-swimming larvae. So in the very early stages, they're free-swimming, move around the ocean, and then they'll settle in one location and become sessile. Sessile means that they don't move. They're rooted to the spot. So they're sessile as adults. Moving on to cnidarians. Cnidarians show radial symmetry again. We can cut it in half in pretty much any direction across the top here, and both sides would match. They have 2 different body plan types, and depending on the species, some species will have both the polyp and the medusa body form in their life cycle. Others will just have the polyp or just have the medusa. Kind of depends on the specific species. The main difference is the polyp is going to be attached to the surface and the medusa is free swimming. Another characteristic that we have in the cnidarians is they have stinging cells. And those stinging cells, you find them on the tentacles of our free-swimming jellyfish. They'd be at the top of these tentacles on the polyp as well. These are cells that have a stinging barb-like hook that's packed inside the cell, and then that cell is pressurized with seawater, so it's under a high pressure inside the cell with a trigger. So you need to just brush against that trigger, and the cell will shoot that barb out at a very high velocity. And they use this to catch fish, so that barb shoots out and gets the fish. They oftentimes have some type of poison or venom to tranquilize the fish, and that allows them to eat it. Not a good thing if you're a human swimming in the ocean and you get hit with some of these jellyfish stingers, because it's very, very painful and can be lethal to humans as well. You'll notice part of the members include corals, and corals are currently having some problems because this is what healthy coral look like. It's very colorful, lots of fish and other organisms living off the coral. Well, that color is caused by symbiotic organisms living in and on the coral bodies, and those symbiotic organisms use photosynthesis, so capture energy from the sun and produce sugars with it, and they'll share a little bit of that with the coral. In exchange, they have a nice place to live inside the coral bodies. However, as the temperatures increase, those symbionts, the algae, actually leave or get killed off by the hotter temperatures, and so the bleached coral is a dying coral. So it's no longer got its symbiotic relationship, and very much like we saw with the mycorrhizae trees in the previous chapter, it simply can't survive without that symbiotic partner. Moving on to Platyhelminthes, and these are flatworms, and some of these flatworms have a full digestive system and others don't. They have just a single opening where, so in this one, in our liver fluke, it may just have a mouth at the top and a single opening through the body, basically a sac, and so it eats food and it goes down into the digestive system. However, to eat something else, it has to regurgitate whatever's left over of that food. That can be very wasteful, so you lose a lot of nutrients that you simply don't get a chance to extract. Also, because they're flat, all the tissue layers of the body are packed right down on top of each other. That means the digestive system is then connected to the body movements. So if this fluke had just eaten a big meal and is trying to digest it, if something approaches and tries to attack it and it engages its muscles to swim away quickly, it's likely to eject the food as it uses its muscles to swim away. So that's a problem when you're a flat organism with all those tissue layers stacked right on top of each other. Another representative from this group would be the tapeworm that you find in mammalian intestines, including humans. The tapeworm is actually interesting because it doesn't technically have a full digestive system anymore. Instead, it just absorbs nutrients throughout its body. So it's a long, long, flat body, and it just absorbs nutrition from its host. In the past, they actually used to sell tapeworm eggs as diet pills. And they worked really great. You'd take the eggs, as the tapeworm hatched and grew, they would start absorbing nutrition. So removing calories from the host, which is great. You start losing weight because a tapeworm is eating half of your food. Problem is, then the tapeworm gets too big and takes too much, and it starts causing health problems. So then you have to deal with getting rid of the huge worm in your intestinal tract. So not a recommended way to go on a diet. The annelid group are segmented worms, and you guys have probably all seen earthworms. They'll crawl out on the sidewalk when it rains heavily. And the exterior of the earthworm has ridges or segments. So if you draw the worm, you can see it's got ridges on the outside of the earthworm. Those ridges actually continue through to the inside as well. So there's membranes separating each of these segments, and then the nerve and digestive tract run through those separated sections. This is why if you were to cut an earthworm in half, say if you're fishing with it, the two halves survive for quite a while because of all of these separated sections, but eventually if you've cut a worm in half, it will die. Other things that are segmented If you've ever gotten a leech on you, or if you're at the river or lakes this summer and you get a leech on you, take a look real close at it, and those leeches are going to be segmented worms as well.